we are studying biodiversity, the biodiversity of mushrooms in Enugu State. And that requires that we gather as many mushrooms as possible and then use a DNA barcode technique to characterize these mushrooms. Right now, I think we have about 89 accessions collected by different students, and we have extracted the DNA. And we're actually planning to do a PCR, polymerase chain reaction. And these things are done by students, by themselves. They have to get their hands wet. So this is not me talking for 2 hours and telling them to go and write me an essay, to answer a question or two, or do an exam for me. This is them now taking pipettes to do what they have to do, and then go ahead to analyze the sequences, the nucleotide sequences that are going to be sent back to us, and then be able to determine the species, the classification of these mushrooms.
Currently, we are running the second batch of the mushroom project. Today, we did DNA extraction using silica resin protocol. The whole mushroom project is more of we trying to understand what the mushroom is about and then trying to have a database of Nigerian mushrooms in comparison with the rest of the world.
We are hosting a DNA barcoding workshop. Well, the theme is biodiversity study for mushrooms, fungi family. We have been able to move from humans, animals, now we are moving into microorganisms and of course fungal family. So today we are studying all kinds of mushrooms. We've already started with the students of Godfrey Okoye University who came earlier. Currently we have students from Ebonyi State. We're going to be hosting people. Our plan is to be able to at least survey the biodiversity of mushrooms from the southeast. So we've done Enugu, we are now doing Ebonyi State. We'll move on to Imo State, Abia State, and of course Anambra State. Yeah.
I'm a student of Godfrey Okoye University, Ebonyi State. Over here, we came for a workshop for DNA barcoding. We are to use a sample of a fungi, which is mushroom. In the process, we got to do DNA extraction by a series of processes, starting off with the lysis of the cell wall to rupture the cell so you can get the DNA down to the last section where we got to extract the DNA after washing it off with ice buffer and TE. Now, I'm very happy for this experience because it gave me a one-on-one interaction with the tutor that helped me understand the practical aspects towards the theoretical knowledge that have been imbibed in us right in school. And I'm grateful for the opportunity to be here and be able to expand my knowledge. And I feel that this would help in my career in gene therapy by getting to analyze the DNA gotten from different samples. I might be able to analyze and check the differences or similarities between the DNA of a particular sample and other DNA, and also how drugs that are synthesized will be able to affect the molecular aspect of living cells. Thank you very much, DNALC Nigeria, for this wonderful opportunity.
My name is Okeze Chamaka Stella, a student of Godfrey Okoye University in Dufa-Alike-Iko, Ebonyi State. I am here at DNALC for DNA barcoding. But one of the processes that we have done today is DNA extraction, and it has enabled me to gain more knowledge. I learned the basic principles in DNA extraction, and I was able to see how we can extract the DNA of fungi, which is mushroom that we use today. I am grateful to DNALC for this opportunity, and I pray and hope that this organization expands to greater heights.
So that's why we exist as a center. We are here to do. We are here to provide evidence. We are here to teach students how to research and how to answer real-life questions that are of importance to society, not just memorizing things that they vomit during the exams and they say they have passed. And then they get out there, they can't get jobs. And you even give them jobs and they can't perform in the jobs.